Good morning, student. In the previous class, we will discuss about meiosis first. Within that meiosis first, we will study about the prophase first. Then, after the prophase first, then we will see the which one? Metaphase first one is there. Okay. In the meiosis, that one is called as reductional division. Meiosis first, that one is called what? Reductional division. Then in the metaphase, here. Homologous chromosome. You will see here. These are the homologous chromosomes are there. Already I have told you about what we mean by homologous chromosome. Those homologous chromosomes, they will come towards the center of the nucleus, or they will align at the center of that nucleus, or it may be they will reach the equator of the nucleus. These are in the what? Pair. Already they will exchange the genetic material in the prophase. Then here they will carry the exchange of genetic material, that is the recombination of the DNA. Then they will arrange in the equatorial plane in the tetrad forms are there. You will see here, these two chromosomes, these are not two chromosomes, these are how much chromosomes are there? 4 chromosomes are there. Within this pair, we want to see what? Double DNA, that is 2 chromosomes are there. Then this one red. Then that one, it is not a single chromosome. It also has what? Double DNA. Hence it is also what? 2 chromosomes are there. Hence those those are comes towards the equatorial plane. Here the chromosome, they will facing opposite to the poles are there. Here this chromosome will facing towards the one end, and this chromosome will facing towards the other end. Here in the metaphase, there is a formation of what? Continuous fibers are there. Those are origin from the which one? Astrons from one end to the other end. That was what? Continuous spindle fiber, which was spindle fibers. Then the next one, chromosomal spindle fibers will be arise in the metaphase. That is what? Those spindle fiber, they will carry the which one? Homologous chromosome to the astron. That is called what? Chromosomal spindle fiber. This is about the metaphase. Only one thing, that is what? Tetrads or homologous chromosome, they will come towards the equatorial plane is there. That is the main part of the metaphase. Then the next one, anaphase. Normally you will know in the mitosis, during the anaphase, the chromosome, they will come towards the 2 opposite poles. Here once again, we will see, those which are tetrads, chromosome, those chromosomes, they will separate from each other. Here, this chromosome, it will separate from this one. This chromosome, it will separate from this one. Then, here the pairing of chromosome, they will become what? Separated. Here, 2 pairs are present at the equatorial region in the metaphase. But here, in the anaphase, they will become what? Separate from one another. Then there is no intracellular fiber. But in the mitosis, in between the 2 homologous chromosomes, there we will see what? In between the chromosome, intracellular spindle fibers will be origin. But in the which one? Meiosis first, anaphase first. Here we can't see the interzonal spindle fiber. Okay. Then the no splitting of centromere. Here the centromere, it is not undergo what? Splitting. Hence here there is no which one? Splitting of what? Centromere. Then the homologous chromosome, they will move towards the 2 opposite folds by the force of what? Chromosomal spindle fiber. Then this chromosomal spindle fiber, it will carry the one one homologous chromosome, one chromosome towards the one fold, and this this chromosomal spindle fiber will carry towards the another fold. Hence in the anaphase first of meiosis 1, here we want to see what? Homologous chromosome, they will separate from one another and they will move towards the 2 opposite poles. Not they will group towards the opposite poles. In the which one? Anaphase, we will see that one. Then the last one, finally, a later anaphase, there we will see at each pole they having what? Chromosomes. Early phase, they will move to early anaphase, the chromosome, they will move towards the 2 opposite poles. But in the later anaphase, the which one? the chromosome, they will group towards the two opposite poles are there. Okay, then the last one, that is called telophase first. Okay, here nuclear membrane, already in the prophase, nuclear membrane, nucleolus, endoplasmic membrane, then as well as Golgi apparatus membrane. These are what? Disappear. But in the telophase first, nuclear membrane, nucleolus, as well as endoplasmic, then Golgi apparatus, they will what? Reappears in the telophase first. Okay. Then spindle fiber, they will become what? Disappear. Here the spindle fibers will be there, but in the telophase, spindle fibers will be disappear. Then the chromosome, they can do what? Decondensation. They will become what? Chromatin threads. Here you will see the chromosomes are there. They will become what? Decondensation. They will become thread-like structures in the telophase first. Okay. Then uncoiling of the chromosome, they will form what? Chromatin network in the telophase. Then spindle fiber disappear. Here we can't see. Then I think which one? Two daughter nuclei they will be formed. Here you will get one in the telophase first. Two daughter nuclei. They having what? Network of chromatin threads in the telophase. Then here after the telophase, it can undergo what? Cytokinesis is there. What do you mean by cytokinesis? Division of cytoplasm. Here you will see the cytoplasm is there. Then in the animal cell, which one? Membrane of the cell, it will form a furrow within the cytoplasm. Then this will be complete. I think it will be continuous. It will become what? Continuous. Then here we will get what? Two daughter cells will be origin at the cytokinesis. But before I think cytokinesis, they can do, after the cytokinesis, finally after the cytokinesis we will get one? Two daughter cells will be origin. Here after the cytokinesis, here we will get the two daughter cells. Then these two daughter cells, they will enter into the meiosis second is there. Before the entry in the meiosis second, these two daughter cells, they want to enter within the meiosis second. Before that one, they can undergo once again, which one? Interkinesis. These two cells, they can undergo G1 phase, they can undergo G2 phase, they can't undergo S phase. These two cells, they can't undergo S phase. This is about the which one? Meiosis part. Then here I want to show once again. Here we will see the homologous chromosome. Within a single chromosome, we will see what? Two chromosomes are there. There is an exchange of genetic material. Here one is completely red and the one is white. Within that red, you will see the exchange of genetic material at the tip of the arm. Here black spot, it is a combination of what? Gene. It will be added in this chromosome. Then within this white chromosome, that will carry what? Red chromosome. It means what? I want to just mention it. Here, recombinant chromosomes will be origin. Okay. Then after when these two cells, they will enter into the meiosis 2. First, within this meiosis 2, once again we will see the prophase 2. Metaphase 2, Anaphase 2, and finally telophase 2. Then here division in the mitosis as well as in the meiosis first, the division it will occurs, I think, longitudinally. But in the which one? Meiosis 2, vertical division will be happens. Okay. Then here we will see what? Both centroids, these are appears towards the two ends are there. Okay. But in which one? Meiosis prophase 2, centrioles, the division of nucleus. This one is okay. The division of nucleus. It will be here we will show the centrioles at the two opposite poles. Okay. Then prophase 2. Once again within the prophase what happens? Nuclear membrane, nucleolus, endoplasmic, then Golgi apparatus membrane. They will what? Disappear. Then here formation of what? Spindle fiber will be origin as well as S star. S star will be origin. What will be S stars? Here we will see the small rays are there around the centriole. This one is a centriole, black spot. That one is what? Centriole. Around this one, it will give rise to what? Small rays from the centriole. The combination of A star rays plus centriole, that combination is called what? Which one? A stars. Okay. Then here the spindle fiber as well as A star, they will appear once again. Then chromatin fiber, once again they can undergo what? Condensation. Okay. They will condensation as well as spiralization. Okay. Then they will become thick as well as short. Here we will get the thick and short chromosome will be origin. Then centriole, they will move towards the two opposite poles. Here one centriole in the telophase first, that will be form another centriole. Then these two centrioles, they will move towards the two opposite poles in the prophase 2. As well as this is another daughter cell. Here we will see the two centrioles will be moved towards the two opposite poles, or you can call it as what? A star. Then after the prophase second, then once again we will see the metaphase. Here I want to draw only this daughter cell. Then you can imagine also here. Okay. Then here the chromosome, they will move here at the equatorial plane of the nucleus. Okay. Here the chromosome, they will move on the equatorial plane. They will become at the center of the nucleus. Then the spindle fiber, they will attach to the centromere. There are two types of which one? Spindle fibers will be origin in the metaphase. First one is what? Continuous spindle fiber. That is origin from one end to other end. But here small spindle fibers will be origin up to here, up to the chromosome. Okay. Then I think spindle fiber, they will attach to the centromere. Here you will see the centromere is there. Then this spindle fiber, it will attach to the centromere of the chromosome is there. Okay. Then this centromere, it will attach to the another chromosomal spindle fiber. Okay. Then here the spindle fiber, they are attached to the centromere. Then after when these chromosomes, here you will see the, I think which one? Homologous chromosome. Here red chromosomes are there. Then they having what? Recombinant DNA in the black spot. Then as well as here we will see the white chromosome. They having what? Red recombinant DNA. Yeah. Then it will undergo what? Anaphase second is there. Then during the anaphase, here I want to mention once again only one cell. Okay. One nucleus or maybe cell. Okay. Here we will discuss about what? Cytokinesis. Hence these are the nucleus. Finally we will call them as what? Cell. Okay. Then here centromere, it will become split up. Here centromere, it will become what? Split. Yeah, it will become what? Split. You must see here. It will become split here. Yeah. Then red with black spot, red with black spot. They will move towards the two opposite poles. Here you will see that one is red with black, red with black. They will move towards the two opposite poles, as well as white plus red. Then centromere will be divided. Then these two chromosomes, they will move towards the two opposite poles in the anaphase second. Sir, how they will move towards opposite poles? Because there is a formation of, in between the two chromosomes, there is a formation of what? In between the two chromosomes, there is a formation of what? Interzonal spindle fiber will be origin. Then in between these two chromosomes, there is a formation of what? Interzonal membrane will be origin. Then they will move towards the two opposite poles. Hence here interzonal membrane will be formed. Then the sister chromatids. Normally we will know about the sister chromatids. Red, these are what? Sister chromatids. Yeah. Then red as well as white, these are what? Non-sister. But these two sister chromatids, they will separate from one another. Then they will move towards the two opposite poles. Now they will become what? Haploid as well as haploid, haploid, haploid. Then finally, after this one, Anaphase 2, then finally we will reach the final stage that is called as a telophase is there. Then here I want to mention this cell, here I want to mention this cell, or it may be nucleus is there. Then finally in the telophase, nuclear membrane, nucleolus, endocorticulum as well as Golgi apparatus, they will be origin once again. They will be formed in the telophase. Then spindle fibers, as stars, they will become what? Disappear. Then finally chromosome, they will become what? Uncoiling. This chromosome, it will become what? Uncoiling. This chromosome, it will become what? Uncoiling. They will become what? Chromatin threads. As well as here, this chromosome, it will become what? Uncoiling. It will become threadless structure. Then this chromosome also, it will become what? Threadless structure. Threadless structure. Threadless structure. Yeah, finally the chromosome they can undergo what? These in the Anaphase 2, whatever we want to see the chromosome, they can undergo uncoiling. They will become what? Uncoiling, and they will become what? Chromatin threads in the telophase. Finally, we will get how much nucleus? Within this single cell, we will get what? 2 nucleus. Within this single cell, we will get what? 2 nucleus. Here nuclear membrane will be reappear, nucleolus will be reappear. Here I am not mentioned the nucleolus, okay? Nuclear membrane will be reappear. Here finally we will get the 4 nucleus. Then these I think these two as well as these 2 nucleus, they can undergo once again cytokinesis. What do you mean by cytokinesis? Once again, fusion of cytoplasm will be happens. Then finally, and furrow, it will be origin in the membrane of this cell. It will continuous. It will be continuous. It will be continuous here, and furrow will be origin in between the 2 nucleus. Then here finally we will get 4 daughter cells will be origin. They having each and everyone having its own nucleus as well as chromatin threads. Then finally in the meiosis 2 of which one? Cytokinesis 2, finally we will get the 4 daughter cells, and these 4 daughter cells, are they haploid or diploid? They will become what? Haploid, haploid, as well as this one also what? Haploid, haploid. This is about the meiosis second. Okay. Then next class we will discuss another concept. Okay. Thank you.